Okay, welcome to today's tutorial. In this video, we're going to look at the kingdom Monera, right? So the kingdom Monera comprises of unicellular prokaryotic organisms that lack membrane-bound organelles, right? And the membrane-bound organelles that they lack include the nucleus, the mitochondria, endoplasmic reticulum, and then the lysosome, right? So the kingdom Monera comprises of unicellular organisms. By unicellular, we mean that these organisms are made up of one cell. They are prokaryotic organisms as well. Pro means primitive, karyote means nucleus. So they have a primitive nucleus. In other words, they don't have nucleus. So prokaryotic organisms means that they lack membrane-bound organelles, right? Or they lack a true nucleus. And some of the membrane-bound organelles that they lack include the nucleus itself, mitochondria, endoplasmic reticulum, then the lysosome, right? So the kingdom Monera, uh, the group of organisms, or is a kingdom that comprises of unicellular prokaryotic organisms that lack membrane-bound organelles. And the membrane-bound organelles that they lack include the nucleus, mitochondria, endoplasmic reticulum, and the lysosome. So having known that, here we're looking at the classification of the kingdom Monera, right? So the kingdom Monera is classified into two sub-kingdoms. These are the two sub-kingdoms, right? They are classified into two sub-kingdoms. We have the sub-kingdom Archaebacteria and the sub-kingdom Eubacteria. So these are the two major sub-kingdoms under the kingdom Monera. So here we're looking at the characteristics of the kingdom Monera. So here, organisms in this kingdom, they have a rigid cell wall. Their cell wall is rigid, right? And like I said before, there are two subkingdoms under the kingdom Monera. We have subkingdom Eubacteria and subkingdom Archaebacteria. So for the subkingdom Eubacteria, their cell wall is rigid due to a substance known as peptidoglycan, right? Their cell wall is made up of a substance called peptidoglycan. That is what gives rise to the rigidity of their cell wall. For the archaebacteria, their cell wall is made up of pseudopeptidoglycan. Pseudopeptidoglycan, right? And this is the substance that is responsible for the rigidity of their cell wall. So these peptidoglycan and pseudopeptidoglycan, they are polysaccharides and proteins. They are carbohydrates and proteins. Peptido symbolizes protein, glycan symbolizes carbohydrate. So it's just a combination of polysaccharides and what? And proteins. Then also their cell membrane, the cell membrane of the organisms in kingdom Monera, they are made up of phospholipids, right? In the subkingdom Eubacteria, the phospholipids are bound together by ester bonds, while in the Archaebacteria and the subkingdom Archaebacteria, the phospholipids are bound together by ether bonds. So note these two differences. In Eubacteria, the bonds that binds the phospholipids that makes up the cell membrane are the ester bonds, while in Archaebacteria is the ether bonds, right? Also, since the kingdom Monera, they don't have membrane-bound organelles, that means they don't have nucleus. And the life of a cell is in a nucleus. So since they don't have nucleus, what they have is the nucleoid. It's called nucleoid, right? So the kingdom Monera have nucleoid, and this nucleoid is an aggregation of DNA materials. So this is the nucleoid of the cell. Nucleoid, right? It is a cluster or an aggregation of DNA materials. The DNA materials in a cell, in a typical cell, is supposed to be centered or enclosed inside the nucleus. But in the kingdom Monera, they don't have a true nucleus, right? They have no nucleus. So what they have is a nucleoid. And this nucleoid is an aggregation, is a collection of DNA materials. Ordinarily, the DNA in the kingdom Monera, they are scattered all over the cell, right? They are scattered all over the cell. DNA materials are scattered all over the cell. So this is a DNA material. This is a DNA material. This is also another DNA material. This is a DNA material. So When you have a collection of this DNA material together, what we refer to that as a nucleoid. So what the kingdom Monera have is a nucleoid and not a nucleus, right? So another important feature of the kingdom Monera is that they have extra DNA structures, and these extra DNA structures are called plasmids, right? So the kingdom Monera, the cells of the kingdom Monera, they have an extra DNA structure called plasmids. And these plasmids, they contain different variety of genes. For example, they contain the F gene called the fertility gene. And this gene will determine if the strain is a positive strain or is a negative strain, right? It is called fertility gene, F gene. For a positive strain, they will have what we call uh, sex pilar. So let's say these are the sex pilar, right? For the positive strain, they produce what we call the sex pilar, right? And these sex pilar are responsible for reproduction, for conjugation, right? So this sex pilar helps the positive strain bacteria to bind to the negative strain bacteria so that reproduction can occur or fertilization can occur. So that's what the F gene is all about, right? Then we have the TET gene, and the TET gene, they are responsible for antibiotic resistance, right? When you apply antibiotics to bacteria, antibiotics are supposed to kill bacteria, right? But some bacteria are resistant to some antibiotics, and this is due to TET gene that are produced by the plasmids. Then also, nutrition in bacteria or in the kingdom Monera is by absorption through their cell wall. So they absorb nutrients through their cell wall, and also some of them can carry out photosynthesis. An example are the cyanobacteria. So organisms in the kingdom Monera, which are the bacteria, they are usually non-motile. They can't move, but some of them can move. Those that have flagella are able to move, right? But most of them are non-motile. Then, so the motile ones, they move with the help of flagella, and they play various ecological role in the ecosystem. They act as decomposers. They can decompose organic matter. They also take part in symbiosis, and also they act as, some of them act as pathogens. Majority of them act as pathogens, right? Pathogens, they are agents that causes diseases. So we've looked at the classification of the kingdom Monera. Like I said, the kingdom Monera is classified into two subkingdom. We have the subkingdom Archaebacteria and the subkingdom Eubacteria, right? So let's look at the first one, which is the subkingdom Archaebacteria. So a major feature of the subkingdom Archaebacteria is that the organisms here, they are capable of living in extreme and harsh conditions, right? And like I mentioned before, their cell wall is rigid because it is made up of pseudopeptidoglycan, and this is composed of polysaccharides and an S layer protein, right? So also the surface layer protein offers them chemical and physical protection, right? So here is the classification of the subkingdom Archaebacteria. They are classified into uh, three main divisions. We have division halophiles, we have methanogens, and the thermoacidophiles. So the halophiles, they are capable of surviving in extreme salty area, right? Extreme salty area. The methanogens, they are capable of surviving in marshy areas, right? And can also be found in they can also be found in guts of cow and buffaloes, and they are used in production of methane from waste, and that is why they are called methanogens, right? Then we have thermoacidophiles. They are able to survive in hot sulfur springs. So these bacteria are able to survive at a temperature, some are able to survive a temperature of 90 degrees Celsius, right? and it is impossible for most living things to survive under this condition. So halophiles, they can survive, or they are found in extreme salty area. Methanogens are found in marshy area or the guts of cow and buffalo, and they are used in production of methane from waste. Then the thermoacidophiles are found in hot sulfur springs, and some of them can survive a temperature up to 90 degrees Celsius. So, having on that, let's look at the subkingdom Eubacteria. So, the subkingdom Eubacteria, they are also referred to as the true bacteria. Eu means true, bacteria, right? So, Eubacteria, they are true bacteria. So, their cell wall is rigid and made up of peptidoglycan. We mentioned that before. And the Eubacteria, they exist in various shapes and forms. We have the cocci. Cocci are round bacteria, they are round in shape. Bacilli are rod-like in shape, right? They are rod-like in shape. The vibrio are command-like in shape. They are command-like in shape, something like this, command-like in shape. While the spirillum, they are spirally twisted bacteria. So they are spirally twisted bacteria. So the most important here are the cocci. So the cocci, they exist in various forms too. Micrococci, micro means very tiny, small. Micrococcus or the micrococci bacteria, they are very tiny spherical bacteria, right? Well, we have diplococci. Diplococci, they are cocci that exist in two double, right? They exist in two, right? They are double cocci bacteria. Di means two, right? So Diplococcus, they are bacteria that are raw, they are bacteria that are round and exist in two. Then we have the Streptococci. Streptococci, they are round bacteria that occurs in chains, right? So they just occur in chain. They are round bacteria that exist in chains. While the Staphylococci, they exist in clusters or in groups, just like this, right? So you should know the difference between each of these various forms. Micrococcus bacteria, they are made up of tiny cocci, tiny round bacteria. The micrococcus, the micrococci are the tiny round bacteria. Diplococci, they are round bacteria that exist in two. Streptococci, they are bacteria that exist in chains. They are round bacteria that exist in chains, while the Staphylococci, they are round bacteria that occurs in clusters or in groups, right? So the word coccus is singular, cocci is plural, right? Cocci is plural, coccus is singular. So micrococcus, diplococcus, streptococcus, staphylococcus. Plural will be micrococci, diplococci, streptococci, then staphylococci. So the last thing that we're going to look at in this lecture is reproduction in bacteria. So how do bacteria reproduce? Mainly, basically, they reproduce by binary fission, right? That is, this is a bacteria, it just splits into two. After condensation of the entire content of the bacteria, it splits into two. That splitting into two is called what? The binary fission. And each gives rise to a new bacteria, right? So bacteria reproduce mainly by binary fission. They can also undergo what we call genetic recombination, and this involves exchange of their genetic material. So genetic recombination in bacteria or in the kingdom Monera could be by conjugation, it could be by transformation or by transduction. So by conjugation means that the DNA passes through an extension on the surface of one bacterium, travels to another bacterium. So if this is a bacterium and this is another bacterium, by conjugation we mean that the DNA passes through an extension. This bacterium and this bacteria are going to combine. They are going to combine. There's an extension that will arise from this bacterium and fix to the next, right? So by conjugation, and what facilitates this conjugation is usually the sex pila. And like I said, the sex pila are produced by what? The F gene, right? They are produced by the F gene. So positive strains bacteria, they have this sex pila. Negative strain bacteria, they lack the sex pila. So during conjugation, the sex pila holds these bacteria together, and then DNA materials then pass from maybe the positive strain into the negative strain, and there reproduction occurs, right? Then we have transformation, and in transformation, the bacteria picks up DNA from the environment. Bacterium picks DNA from the environment. Then we have transduction, and transduction involves viruses carrying DNA from one bacterium to another. So when a virus carries DNA material from one bacterium to another, we refer that mode of reproduction or genetic recombination as what? Transduction. So know the difference between conjugation, transformation, and transduction. In conjugation, the DNA passes through an extension on the surface of one bacterium to another, right? So this is one bacterium, this is another bacterium. During conjugation, the DNA material travels from one strain of the bacteria to another. In transformation, the bacterium picks up DNA material from where? The environment. And in transduction, viruses carries DNA from one bacterium to another, right? So let's look at this question. Which of the following involves the transfer of DNA via bacteriophage? So first of all, you must know what is a bacteriophage. Bacteriophage are viruses that infect bacteria. So which of the following involves transfer of DNA via bacteriophage? Via the virus that infect bacteria. So is it binary fission? No. Conjugation? No. Transformation? No. And it's by transduction, right? It's by transduction. So remember what we said, that when it involves a virus carrying DNA from one bacterium to another, we call that transduction. When it involves bacteria picking up the DNA material from the environment, we call that transformation. And when the DNA material passes from one bacterium to another, just as we described here, from one bacterium to another, we call that what? Conjugation. But the major mode of reproduction in bacterium is by binary fission, right? So with this, we come to the end of today's tutorial. In the next class, we will look at gram staining.